Good evening and welcome back to our classes. This is the third part of the three-part series where we've been discussing the bacterial cell and its structures. Today we are going to discuss the internal structures of the bacterial cell and what they look like and how to identify them and the functions of all these different parts. Now we ended the other video by talking about the cell wall and the cytoplasmic membrane. Now it's good to know that the cytoplasmic membrane is also the first part or the first internal structure of the bacterial cell. And like we said before, the cytoplasmic membrane is made up of a phospholipid bilayer. Now, it's easy to break down and understand this. Phospholipid talks about phosphate and lipid. So we know that the layer has a phosphate head which is hydrophilic. Hydrophilic meaning that that phosphate head is water loving. And then you have hydrophobic tails which are made up of glycerol. So the cell wall is made up of this phospholipid bilayer. It has the phosphate head and the glycerol tails that make up this layer. Now, apart from the cytoplasmic membrane, we have what we refer to as the protoplast. And the protoplast simply means the cytoplasmic membrane together with the cytoplasm. All that structure together within the bacteria is what we refer to as the protoplast. So we have the cytoplasmic membrane, we have the protoplast. Now, when we go further inside, we have the cytoplasm itself. Now, the cytoplasm, some people refer to it as the liquid part of the cell, where a lot of biochemical reactions do take place. But it's good to look at the cytoplasm in three parts. The cytoplasm has three parts. Now, the first part of the cytoplasm is the granular area or the granular region of the cytoplasm. And what is in this granular region? In this granular region, we have a lot of RNA, we have a lot of proteins, we have a lot of other bodies such as Golgi bodies, ribosomes, which are present in this granular area of the cytoplasm. And this is important to note in a bacterial cell. Now, apart from the granular area, the second area is the fluid part of the cytoplasm. The fluid part of the cytoplasm contains a lot of fluid and dissolved substances within the bacterial cell. Now, there are so many dissolved substances that can be in the bacterial cell, such as proteins, such as starch, such as glucose. All of these are dissolved within the bacterial cell. Now, apart from that, the third part of the cytoplasm is the chromatin area, and this refers to that region of the cytoplasm that we also refer to as the nucleoid area, which is rich in DNA and all sorts of genetic material. So this is important to note. Now, when we leave the cytoplasm and go even further deeper, we come into the nucleoid region of the cell. Now, like we've said before, and we'll continue to say, that the bacterial cell contains one single giant chromosome, one circular DNA. I've explained in further videos that you can go and check up, see what it means to have a single circular DNA and see the relationship between DNA and chromosomes. So we have one single giant circular chromosome in a bacterial cell, and this is what we see in the nucleoid region. The nucleoid region of the cell does not have the nucleoid region of the bacterial cell does not have a nuclear membrane. Rather, it's just your DNA material that is within this region, and this is important to know. Now, lastly, it's important to also know that bacteria usually go into spore formation or cyst formation, and this is important. And why does bacteria do this? There are a couple of reasons why bacteria can either go into spore formation or cyst formation. But it's important to know that majorly there are two types of spores that are formed by bacteria. There's the endospores and the exospores. Now, the endospores continue to remain within the bacteria until, and then why would the bacteria go into spore formation? Bacteria goes into spore formation if environmental conditions become harsh and the bacteria finds it hard to survive. Now, in order to conserve itself and preserve itself over this harsh period of time, the bacteria can go into a hibernative stage. And then when it goes into this hibernative stage, or sometimes referred to as a vegetative state, the bacteria now breaks down and then forms spores. But these spores are within the mother cell and they stay there. We refer to them as endospores, and they remain there until conditions become favorable, and then they are released into the atmosphere and germinate and go back to being bacterial cells. Now, there will be many cells that will come out of that single mother cell. That is for endospore formation. Also, some bacteria form exospores, and these exospores are little spore structures that can be found on the surfaces of such bacteria. And bacteria usually do that as a means of dispersal of some of their DNA and their genetic material so that new organisms can be formed. So bacteria form exospores and they also form endospores. So to do a quick roundup of the internal structures of the bacterial cell, we can look at the cytoplasmic membrane, protoplast, then cytoplasm, and then the nucleoid region, and lastly we discussed cyst or spore formation. Thank you for listening to this video and have a wonderful day.